speakers today. Before we start today's events, I would like to refer you to the Qatar University Health CPD website. We have an exciting series coming up following this research series. At the end of this month, we'll be starting our antimicrobial stewardship 6-part series, in a collaboration with Hamad Medical Corporation and Qatar University experts to deliver some education and training around antimicrobial stewardship. So this is something to look forward to at the end of the month. But before that, after today's session, we'll be starting our qualitative research skills series. We have 2 sessions that again focus on the appraisal and design of qualitative studies. So please refer to our website, follow us on Twitter via QU Health, and you can receive all the latest updates regarding our activities. So, as always, we welcome your feedback. So, following today's event, please feel free to contact us either by email or via our Twitter feed, and pass on your feedback to us. So, to introduce you to today's event, it's focusing on the appraisal and design of randomized control trials. We have an expert panel with us today. We have Dr. Prem Chandra, an academic research scientist from HMC, and we have Dr. Mohammed Yassin, a consultant hematologist also from HMC. So I'd like to welcome our speakers and thank you for their efforts in developing the content for today. And at this point, I will now be handing the virtual mic over to Dr. Mohammed Yassin, who will be starting off today's presentation. Dr. Mohammed Yassin, I've now made you the presenter. You should be able to share your slides with the participants.
Assalamu alaikum and good evening, everybody. Welcome everybody to this session of clinical trials. We are going to talk about 1 hour, and I'm trying to focus on the terminology of clinical trials and try to make you aware about the basic concepts of clinical trials and the rationale behind clinical trial so that we can go smoothly to the next practical session. So we would like to start this session by a question, and I would like you to answer these questions and to vote. What are the similarities between clinical research and medical practice? So number 1 is both of them provide care to patient and subject. Number 2, follow treatment regimen exactly as defined in protocol for all patient subjects. Number 3, oversee staff of medical professionals. Number 4, treat patient subject based on individual needs, based on education and experience. Number 5, collect data for all patients subjects using a consistent and specific format. So the 2 similarities between the clinical practice and medical research. So the 2 similarities between the clinical practice and medical research is the follow-up. Number 1 is that both of them, they are providing a quality care to patients and subjects, and both of them also oversees staff of medical professionals. If we go to point number 2, which is follow treatment regimen exactly as defined in protocol for all patient and subject, this is very specific to clinical research, because in clinical research or in clinical trials, you have an inclusion and exclusion criteria, which is clearly demarcated in the protocol, and you have to follow it exactly. So there should be no deviation usually from the protocol. And then the last point, which is collecting data or patient using a consistent and specific format. This is, we call it a CRF, or clinical research form or data collection form. And we use this in clinical research, where you use a unified data set for all of our patients. So if we want to summarize this initial slides, we can say that in medical practice is based on individual needs, and it is conducted with a proven marketed drugs, devices, or other therapies. So for the first point, based on individual needs, you know that we are in the era of personalized medicine, which each category of disease is personalized, and each patient is looked for as a unique patient. So you have to tailor your treatment according to the patient. And the second point, that it's conducted with a proven marketed drugs, devices, or other therapy. As you know, in clinical practice, we are always referring to the evidence-based medicine, and we are looking to the levels of evidence, and we are using approved marketed drugs or devices. Usually, we refrain from using the off-label therapies, and we don't use the therapies which were not approved by approval agencies. While in clinical research treatment regimen, it's based on protocol, and the protocol having a many component. Among them, as I said, is inclusion and exclusion, and it has to be followed as it is. It must be strictly followed by all subjects. It contains an experimental intervention to test a hypothesis, and it may include a placebo. So coming back to our main topic, how we define a clinical trial. Clinical trials usually are defined as scientific investigations that are prospectively examine and evaluate the safety and efficacy of a new drugs or therapeutics using a human subjects. So if you look at these definitions, number one, it's a scientific investigations because it's a research. So we are researching something, and it has to be prospectively examined. This means we are not doing a retrospective clinical trial. When we talk about a clinical trial, we talk about a prospective examination. We always cares about safety and efficacy of the medications because we are dealing with human subject. So when dealing with human subject, safety is coming first. And then we also care about efficacy of those medications. So we want to use a medications which can give a good effect on our patient, a good therapeutic effect on our patients. And we deal with the human subjects. So this is a human subject research, and there is a lot of issues with regard to the human subject research. The result that the studies generate are considered to be the most valued data in the era of evidence-based medicine. Classically, when we talk about clinical trial, people think about medications, drug A versus drug B, but clinical trials, this is part of clinical trials or type of clinical trial, but clinical trial is not limited to drug therapy, is not limited to medications. It can be about drug therapy, as you all know, that you can compare 2 or 3 or whatever number of medications in clinical trial. It can be about a medical procedure. And I will give you an example of a medical procedure, a very famous trial, which is looking for primary angioplasty. You know, patients with coronary artery disease, they can present with angina, or they can present with acute coronary syndrome or myocardial infarction, and they have a blockage for one of their arteries in the heart. And one of the studies is trying to evaluate whether intervention with primary angioplasty means inserting a stent to try to overcome that closure mechanically will be useful or not. So in this condition, the intervention is using a mechanical stent. A mechanical stent here is a procedure because you are going to do an angiography, and then you are going to push this stent and you reopen this closed vessel. Another one is educational intervention. And example for educational intervention is one of the very famous trial which was conducted in Zimbabwe, which is one of the countries in Africa where HIV is quite common. And one of the causes for why it's so common was because maybe lack of health educations, and the teenagers and partners, they are not aware about the safe education, about the how to practice safely. So the investigators try to categorize the group into like 2 groups. They gather the group of 2 teenagers, an adolescent and adult, and they divided them into group who received a structured education. It means they educate them about the sexually transmitted diseases and how to prevent themselves about the sexually transmitted diseases. And the other group who didn't receive the same quality of education, and they notice the difference. So this is one of the examples where you can use an education can be a clinical trial, and you can have like two arms of educations or even more. One group is educated and the other is not educated, or you can have a structured versus unstructured education. And I give an HIV as an example, but there are many trials in educations in smoking, which there's a lot of studies about smoking and others which talk about education as intervention. The screening trial, maybe the famous screening trial which was started in the late 50s, is UK screening for UK screening for endothelial cancers in female. Because female, they can get some sort of gynecological cancer, and the pap smear was just invented. So they used the pap smear as screening for cervical cancer in female. And this is an example where you use the intervention, and the intervention here is using the pap smear to look whether it's positive or negative, and this can give you a clue whether the lady is susceptible to have a cervical cancer or not. So here we are screening for a cervical cancer by using a specific technique. A vaccine trial, maybe one of the famous vaccine trial is Manchester trial. Manchester trial is a trial which was conducted in Jamaica, and we know that Jamaica is a country which is prevalent of sickle cell disease, and the cause of morbidity and mortality in patients with sickle cell disease is acute chest syndrome. And acute chest syndrome is equivalent to pneumonia in most of the definitions and most of the references. So it was noticed by investigators in Jamaica that most of the children, they die at the age of 2, and the cause of death usually is related to pulmonary complications. So they thought, if we give those children a vaccine, this vaccine may prevent the death or reduce the complication. So they invented the vaccine, the pneumococcal vaccine, and after that, different version of this vaccine, the 7-valent and the 23-valent, and it proved that intervention by vaccination in a setting of clinical trial is quite useful, and that one became a standard worldwide, and it saves millions of lives worldwide because we use the clinical trial in academic interventions, and a lot of lives were saved because of that. So clinical trials are not only about medications. It can be about medical procedure, education, screening, and as we explained, all of this. We always talk about randomized controlled trials. So does the trial always have to be randomized? And when we talk about randomized controlled, do we have uncontrolled trials? And if we have the uncontrolled trials, why we should have uncontrolled trials where we have now we are talking about the gold standard, which is randomized, blinded clinical trials. The uncontrolled clinical trials are still exist, and these studies, we use it frequently when a new treatment are studied in the absence of a control group. And as a result of these studies provide less information on the therapy than controlled trials. So what is the necessity? Why we should use uncontrolled clinical trials? Is there is a necessity for this? The answer is yes, and I gave 3 examples. Number 1, maybe there is ethical consideration to do control, to do like a control trial, a randomized control trial, or randomize the patients. And a clear example of ethical consideration is a surgical procedure. If somebody is doing a clinical trial from surgical team, and this clinical trial on appendectomy, it means patient is coming with acute appendicitis, and they have to be taken for appendectomy. To relieve them, nobody can say and come and say, no, it's better to randomize these patients into 2 groups, a group which is going to be to undergo appendectomy, and the other group no appendectomy, because if the surgery is a necessity and the appendix is so, in the late stage, if you didn't do the surgery at that stage, maybe it will rupture and the patient will die. So in some surgical procedure, it's not possible to do the randomization. So we are talking about randomization as a gold standard, but it's not always possible. An example of this is surgical procedure, where sometimes it's a must to have a surgery, and there is no other option apart from surgery. The second common example is if you want to generate a new hypothesis for further research. If you want to generate a hypothesis, usually in the evidence is coming from observations. And observations usually are generated from case report, case series, and then we can go to pilot study. So the case, when you have a case report or a case series, you have a small number, and you are at the stage of testing the hypothesis. So you cannot randomize people or do a blinding before you test your hypothesis at a smaller scale and make sure that what you are testing is really true. So uncontrolled trial is used in generating a new hypothesis for a further research. This is another example. And then the third example where there is a rationale for performing an uncontrolled trial is when you are talking about the pharmacokinetics. You want to investigate the pharmacokinetics properties of a novel drug through a phase 1 or phase 2 clinical trials. You want just to see how the drug is absorbed, how the drug is metabolized in the human body, how the drug is eliminated in the human body. Here, you don't need to do a randomization. You can do uncontrolled. You can do it in one group of participants. So these are three rationales for why we're still doing an uncontrolled clinical trials in the era of randomized controlled trials. And so what are the limitations for these uncontrolled trials? As I said, there is a necessity to do it in certain situations, but we have to be aware that there is some limitations with this. I mentioned three of them. Number one, it's susceptible to investigator bias because there is no randomly selected groups for comparison. Number two, there is a susceptibility to over-interpretation of the result because usually when you are testing a new hypothesis as an investigator, you will be so excited to get a positive result and you may over-interpret or magnify your result. The third one is it's much less informative than any other studies have a concurrent non-randomized control group. We sometimes, as a synonymous, we are talking about a quasi-experimental design. So quasi means like true, and or some people use it as a synonymous for uncontrolled trials. So what is the differences between the true experiment, which we were just talking about, that there is a true experiment and there is uncontrolled experiment or quasi-experimental design? Just to give you an overview, in the true experiment, randomization is usually there. So you will randomize your patients. While in quasi-experimental design, usually it's a non-random. The second difference is in the design of treatment. In the true experiment, you are designing the treatment. I'm going to start the medication A with a dose of 25 milligram for 1 week, and then I will give a maintenance dose of 50 milligram, and the patient will continue on this 50 milligram for like a year. While in quasi-experiment, you don't have to have a control over treatment. You don't have a control over treatment. Maybe the same group of patients treated by many physicians, and many physicians individualize the treatment for those patients. So you don't have an upper hand on controlling the treatment. And then in the control group, in true experiment, usually there is a control and treatment group. And while in quasi-experiment, there is a control group are not required, but it may be used. So we are talking frequently now about blinding and randomization. So I'm going to give you a set of questions. Just think of it and try to figure it out rapidly. So we select the best answer for this cues. Number one, randomization and blinding refer to the same process. Number two, randomization means masking of treatment. Number three, the process of assignment of trial subject to treatment called blinding. Number four, if randomization combined with a blinding, this will increase the bias in clinical clinical trial. Number 5, blinding can be single or double. Can you select the best answer, please?
Dr. Hamid Yassin, the polling should appear on the right-hand side. So participants have about 10 seconds left to register their answer, and then I can give you a summary of their answers.
Yeah, okay. I'm just waiting for the pause.
Okay, so the poll is now shut and is summarizing results. In about 10 seconds, I can give you the scores of the participants. You should be able to see the results on your screen.
Okay, so none of the participants replied that blinding can be single or double. None of the participants answered A, so A we have 0 answer. For B we have one person answer that is correct. For C we have 2 person answer that it's correct. For D we have 2 persons answer that's correct. But majority of people answer the correct answer, which is blinding can be single or double. So when we talk about clinical trial, one of the main features in clinical trials and the landmark for clinical trials are blinding and randomizations. And we are doing a blinding and randomizations to eliminate the bias in clinical trials and to have a scientific publication with a scientific integrity. So as you know, bias is any trend in the collection, analysis, interpretation, or publications, or review of data that can lead to conclusions that are systemically different from the truth. And randomization and blinding are 2 techniques used to control the bias in clinical trials. So what do we mean by blinding? When we talk about blinding, I think even in the layman term, blind means you don't see or you mask. And in clinical trial, it means masking or blinding or masking is a procedure in which one or more parties to the trials are kept unaware of the treatment assignment. This one party can be the investigator. So the investigator may be blinded. He doesn't know the patient are randomly assigned to different ways, and he doesn't know whether the patient is taking the real medication or he's taking the placebo, for example. So, and what randomization means? Randomization is the process of assigning trial subject to treatment or a control group using an element of chance to determine the treatment assignment with randomization. Each subject is randomly assigned either to the experimental group, which receives the investigational manufactured product or IMP, or the control group, which typically receive either a placebo or the comparator group. So this is how we define the blinding and randomizations. We have a different types of blinding. We have a single blinding. We have also open label. We have a double dummy. We have a double blind. Just I will give you an example about 2 of these. What does it mean? And you can, if you want to know more, you can go rapidly through the slides. I make the connections. So when we talk about open label, this means the identity of the treatment assignment is known by the sponsor, the investigator, and the subject. And the name itself is self-explanatory. It's open label. It means everybody can see it. Everybody can know about it. When we talk about single blind, this means the sponsor, there is someone who is blinded to the treatment. The sponsor and the investigator know the treatment assignment, but the subject do not. This is single blind. And then we have definitions for what is double blind and what is double dummy. But we have a different types of blinding in clinical trials. Can we do unblinding if I am doing a single blind or double blind randomized controlled trials? Can I do and blinding in the case of emergency. Assume that patient A is taking an investigator product, and this patient came to the emergency with a heart rate of 150 and shortness of breath. Yes, emergency unblinding of the medications is allowed in the setting of clinical trials, but it has specific regulations and a specific way of documentation. But there is what is called emergency unblinding. It means there is a process to reveal the identity of the investigational product if there is an emergency happens to the patients. Because in the beginning of this trial, of this lecture, we said that the safety and efficacy. So safety always comes first. So why randomization is important, as we said, so that all groups looks alike as possible. And number two, it provides the best way to prove the effectiveness of a new agent or intervention. When we talk about clinical trials, it's of paramount importance to know about GCP, which is referring to good clinical practice. So what is good clinical practice and what are the cores of good clinical practice? We have some tools here. I need you just to answer the questions. Select the best answer out of these 5 answers. Number 1, GCP has nothing to do with ethics. It's only IRB regulations. It means it's only internal regulations for IRB. And we mean by IRB, Institutional Review Board, which in some countries called Ethics Committee. GCP is mainly designed to protect the investigators. GCP is a local regulations only applied to Europe. Data integrity and quality are not part of GCP. Protocol and science are among 13 core principles of GCP. Can you vote, please? Okay, time over. Now we are waiting for the answers. Okay, so one person answered A, which is wrong. Another person answered B, which is also not a correct answer. No person answered C. C also is not a correct answer. And no person answered D, which is not a correct answer. 15 answered E, which is a correct answer. That protocol and science among the 13 core principles of GCP. So frequently we are talking about the ICH GCP. Just to familiarize you with the terminology, what do we mean by ICH GCP? ICH historically refers to International Conference on Harmonizations on Technical Requirement for Registrations of Pharmaceutical for Human Use. So what does this long title means? It means when we are doing a clinical trial, we need to have an official international language for clinical trials. So when we talk about, for example, phase 1, everybody agrees about what is phase 1. When we talk about protocol, all of us, we know what does a protocol means and what are the components of protocol means. When we talk about IRB and regulations, we know what does IRB and regulation means. And to facilitate the GCP, the main aim of the GCP also is to protect the right, safety, and well-being of the trial subject, and to ensure the quality and scientific integrity of the trial data, so that it harmonizes the clinical trials worldwide, and it protects the right and safety as well as well-being of the participant. Classically, the GCP is described as having 13 core principles. And these are the 13 core principles. I will not go through it because of the time. Just you can look at it. It's ethics, and in ethics, clinical trial conduct is consistent with GCP and applicable regulation requirement. do a clinical trials without going through a governance system. This governance system can be in your medical research center, or it can be a regulatory authority in your university or whatever. And then the benefits to conduct a trial justify. So this is a very common principle that when we do a clinical trial, we always look for the benefit and the risk. So the benefit should overweigh the risk. If the risk of overweighing the benefit, this is considered non-ethical, and the clinical trial or the proposal for clinical trial will not obtain the ethical approval. Then another one, just to go rapidly over it, is the informed consent. Informed consent is very important because consent is a legal which govern the relation between the investigator, the hospital, the sponsor, and the patient. And this legal document has to be very clear. It has to be in a language which is spoken by the patient so that the patient can speak it and can understand it and very comprehend it. So if somebody is fluent in Arabic and he doesn't know English, it's not right to consent him on English. Maybe he's good in Arabic, but he doesn't understand. So you should use language. It should be also written very clearly. It should be very clear to the layperson. You should not use a medical jargon, which is very complicated for people to understand. As I said, the consent is mainly directed to the patient, and the patient should understand clearly what is a clinical trial, what is the procedure, what is the risk, what is the benefit, what will happen to his samples, and there is a lot of details with regard to the consent. So this is slide summarizing overall the GCB 13 core principles. When we talk about a protocol is one of the key documents in clinical trial, and it explains how the trial will be conducted, and it serves as a plan to be followed for all those involved in the trial. The protocol include in the trial usually it will consist of an objective or rationale of the trial, why the trial is done and what are the objectives, the primary or secondary objective of the trial, the design of the clinical trial, the methodology, how the clinical trial will be conducted, in statistical considerations, and organizations. It means what will be the organizations, how people will work in these clinical trials, who will lead these clinical trials, who will be the coordinator of these clinical trials, how these clinical trials will be run, in which facility, conducted center, hospitals, will it be in a one center, in a multi-centers, so all the organizations. So these are the overall or general component of the protocol. So clinical trial should be complying with the GCP, which is a good clinical practice, 13 core principles. The clinical trial should have a protocol. The clinical trial should have an approval. And the clinical trial also is frequently subject to monitoring and audit. So when we talk about monitoring and audit, I put these two slides just to make you understanding what is the difference. Monitoring the progress of the trial is the primary methods a sponsor uses to help to ensure that the trial is conducted and documented properly. The sponsor is responsible for developing a risk-based approach. So apart from this long definitions, what does monitoring and audit means and what is the difference between them? When you do a trial, you want to make sure that everything is going right. The selection of the patient is right. The approval, the ethical approval is obtained from the approved authority. The inclusion and exclusion criteria are clearly met, and the consenting is documented in the system where you are doing a clinical trial, and all the logistic around clinical trials are properly. Most of the trials which we will do, it will be sponsored. I mean, it can be sponsored by pharmaceutical companies. And in these occasions, most of the pharmaceutical companies, they are very keen to monitor. It means they are the one looking at all these elements, and they want to make sure that the investigator as well as the hospital or the institute is complying with everything. So monitoring, we can call it as a self-process. It means you don't need usually a monitoring, it's a third party. You monitor what you are doing. And this is with reverse to the audit. Audit, usually you have an independent party, company sponsoring an investigational product or medication, and we have the investigator. For example, Dr. Yasin is doing a clinical trial with X or Y company. This X and Y company, they can send a representative from them just to monitor Dr. Yasin. So this is a monitoring. It's something between the investigator and the monitor and the IRB of that institute. But if we talk about a party which is not related to that pharmaceutical company, and also not related to the investigator or to the institute, and coming as independent party and look for all details, for the protocol, for the ethical approvals, for the compliance. They will review all the consent. They will review the inclusion, exclusion. They make sure that the patient is fulfilling all the criteria. So monitoring is a self, we can call it as a self process, and audit is an external process. So we move again to another question. In this question, we want you to select the correct answer about the clinical trial. In phase 1 clinical trial is concerned about post-marketing. Phase 2 clinical trial is testing the efficacy of the drug on healthy individual. Phase 3 of clinical trial examining the pharmacokinetics. Phase 4 of clinical trial examine only very small number of patients. If phase 3 obtained a positive result, it is called landmarks. Can you please vote? Okay, so the polling is ended. We want to see the result now. Can we have the result, please? Okay, so here we have 5 people answer that B, which is phase 2 clinical trial, testing the effect of the drug on healthy individual. And this is the wrong answer. And then we have 6 of you answered E, which is number 5. If phase 3 obtained a positive result, it's called as a landmark study. Okay, that's fine. So let us move. So pharmaceutical companies usually are dividing the clinical trials. There is different way to define and to classify clinical trials. One of them is to define them by phases: phase 1, phase 2, phase 3, phase 4. So what is going on in phase 1 usually? The manufacturers usually test the effect of the new drug in a healthy volunteers or patients unresponsive to usual therapies. And usually they are looking here for pharmacokinetics and pharmacodynamics. It means they are looking for how the drug is handled in the human body and how it is metabolized and eliminated. So it's mainly healthy volunteers or patients are responsive to therapy. We look for pharmacokinetics or pharmacodynamics. Usually it is a small scale trial, phase 1, and usually it looks also to the immediate short-term safety of higher doses. What about phase 2? Phase 2 examine the dose response curve in patients and what benefit might be seen in a small group of patients with particular disease. So in phase 1, we already tested the pharmacokinetics and pharmacodynamics. Now we want to test the dose. We can use different doses and see which dose is safer, which dose is more beneficial for the patient. So it's mainly a dose response. What about phase 3? Phase 3, usually the new drug is tested in controlled fashion in a large patient population against placebo or standard therapy. And usually phase 3, in a lot of occasions, it will be a multi-center. It will involve a larger number of patients than phase 2 or phase 1 clinical trial. And this is a key phase where the drug will be either will make it or it will break its reputation and with respect to safety and efficacy before marketing begins. And if it is a positive study of phase 3, we call it a landmark study. So landmark study is a positive phase 3 study. So this is the general definitions of the different phases of clinical trials. Phase 4 is the post-marketing. When the drug is marketed, it means the drug is approved by the approved agency, like FDA in US, for example, or EMA in Europe, or European Medical Agency. So the drug is approved, and now the pharmaceutical company is starting to sell the drugs to different countries, and clinicians start to use it. In the setting of clinical trial, we have a selection, we have inclusion, we have exclusion. There is a lot of exclusions for patients with heart failure, with renal failure, with hypersensitivity. So there is a difference between the patient in phase 3 and phase 2 and the real life. So phase 4 is post-marketing. It means it tests the real life scenario for patient, or the patient in the real life, and it also tests the long-term effects of those patients. So also phase 4, which is a post-marketing, it means even after the approval, the drug has to be monitored and adverse events has to be reported. And this is very important because still it can make a lot of differences. So again, we do trial design. Just please select the correct answer. Crossover is the most common design. Pilot studies are obsolete and should not be done. No need for washout in a crossover trial since it has no value. Cluster is done for a small group and not suitable for a large group. None of the above.
Dr. Mohammed, there's one question. Can you describe what washout is, please?
Yes. When we talk about, I will describe it in the coming slide. So when we talk about washout, usually when you use 2 medications, it means I use, for example, in patient chronic myeloid leukemia, we use a medication called hydroxyurea. And then the definite treatment for patient with chronic myeloid leukemia is tyrosine kinase inhibitors like imatinib or nilotinib or dasatinib. So before starting the second drug, I should give a period. This period in most of the medications, it will be like 2 weeks. It means patient should be off any medicine for 2 weeks. So I start the drug A till I achieve the certain target. Then I stop drug A and I'm planning to start drug B. But before starting drug B, I need to give like a period of no medications, like 2 weeks, for example. And we call this a washout period. Okay, so majority of you, so we have 5 answered, none of the above, and we have 5 answered. Crossover is the most common design. Actually, crossover is the most common design. A is the correct answer. So we just described that the clinical trial can be described by phases: phase 1, phase 2, phase 3, phase 4. There is also other types of clinical trial design, and this is a descriptive design. It means the most common of it is called the parallel group. What does a parallel group mean? It means I am testing a 2 medication, just drug A and drug B. Just I will show you this one. So this is a clinical trial parallel group where I will screen my populations, and then I will have a baseline for all of those. I want to make sure that they have a baseline for inclusion and exclusion. And then I will have like a 2 arm A and arm B. Arm A can be the experimental. It means if I have patient with immune thrombocytopenia or ITP, and I want to use a new medication for ITP, say for example L-thrombobag or romoplastin. So this will be the experimental. And then I want to use a control. The control usually we use, if there is a gold standard, we use a gold standard. So you usually test the new medication against the gold standard, for example. The gold standard in ITP is steroid. So I tested against prednisolone or steroids. So I used TPO agonist versus prednisolone, and then I will see the outcome in these two groups. So we call this a parallel group design. This is the commonest design which we use in clinical trial, parallel group. And the patient can be, as I said, randomized to new treatment versus standard treatment or gold standard, or standard treatment versus placebo. There is a different way of randomization, but it's like arms. You have like a parallel arms. It means you have a group in the first arm and another group in the second arm. So the crossover trials randomize patients to different sequences of treatment, but all patients eventually get all treatment in a varying order. The patient is her own controls. We have another type of trials called factorial trial factorial trial. So what do you mean by factorial trial? It means patient can be assigned to more than one treatment or comparison group. And these are randomized in one trial at the same time. It means just to make it easier for you, we will go to the cartoon so that you understand what do we mean by factorial. Okay. So sorry, we talk about the crossover. This is a crossover. Just not to go back. The crossover, it means you have the arm A and you have the arm B, and then it means patient on this arm can go to this arm, and this patient in the other arm can go to other arm. And again, we have the same screening. We have the baseline, and then we start the treatment phase, and then we have the washout. And as I said, the washout, it means if the patient is receiving drug A and I shift him now to drug B, I should keep him off treatment for at least 2 weeks, 2 weeks or 10 days in most of the clinical trials. And same for patient on drug B. If he received drug B and then I want to randomize him to drug A, then I have to do the washout and to move to other one. This kind of trial, we call it crossover design. The third type of design, which we are talking about, is called the factorial design. And in that one, the patient will be designed will have like 4 options. Just to make it simple for you, how the patient will have 4 options in the same trials. Patient can be in group A, this is one option. Then can be in group B, this is a kind of second option. It means group A means he will receive the drug A. Group B means he will receive the drug B. Then patient can be in group AB, it means he will receive drug A and B together. And patient can be in the control group. So you can use the same trial to have like 4 groups. And the advantage of this kind of trial, it has an advantage that you don't need a large number of patients, because you can use the same group to test different types of medications, drug A, drug B, drug A with B, or use a patient in control. So this is a little bit more complicated trial than the classical trials. And usually there is different types of factorials. The most common type is 2 by 2, which I just explained. Other ways of also describing the clinical trials is defining them by superiority, equivalent, and inferiority. So just to make it simple for you, if I told you that I am superior to you, this means what? It means I believe that, and I have an evidence that I am much better than you. And if I told you I am equivalent to you, this means I am as good as you. And if I told you I am not inferior to you, this means at least I am as good as you do. I can do as good as you do. So the clinical trials, we can do superiority studies where we want to prove that drug A is superior to drug B. We can do an equivalent study where we want to prove that drug A and drug B are equivalent in the efficacy. And we can do a non-inferiority study. Usually the non-inferiority study is done to show the effect of new treatment, that the new treatment cannot be said to be significantly weaker than the current treatment. And then, so this is with regard to the superiority. It's superiority study, equivalent study, and non-inferiority study. And I just simplified. I said just take it. I am as good as you. I am better than you, or I am not bad. I can do at least what you can do. This is the in layman terms. Then we have a pilot study. A pilot study is a small study that helps to develop a bigger study. If I have a concept, and this concept is costing $1 million, I cannot go to sponsors or pharmaceutical or to organizations and tell me, give me $10 million to do a clinical trial. They will tell me, do you have a proof of concept? It means, did you do this trial in a small number of patients? And you see, if it works, then we will be more enthusiastic to support you. If it doesn't work, then you call it a day, because even if it doesn't work in a small scale, there is no need to do it in a large scale. This is an example of a pilot study where we are doing one of our clinical trials. We are testing the blood transfusion. You know, there is two types of blood transfusion. One of them is the autologous and the homologous. The autologous means you donate your blood when you are healthy, and then you can take your blood back. And the other type, that we take a blood from somebody who's compatible, his blood is compatible with you, and we give it for you. And this is a very frequent problem among athletes, because it is used to cheat. Because some of the athletes, they will donate their blood, and then they will take it. I mean, they will take autologous blood. And the reason for this is to increase their oxygenation and performance, so that they can do better and they can beat their component. So this is a pilot study where we are going to do in two groups of athletes. We tested the two types of transfusion, and then we use DNA microparticles to see whether these athletes are cheating or not, because it will be sometimes very difficult to know whether the person took his own blood or not. So we have some biomarkers which we are testing to see and to reveal the cheating in such kind of conditions. So we do this at a smaller scale. If this is successful, then we can do it in a larger scale, and we can move to different stages. So in clinical trials also, frequently we are talking about clinical trial endpoint. Always we are talking about primary endpoint, secondary endpoint. So we define the endpoint as a measure that allow us to decide whether the null hypothesis of a clinical trial should be accepted or rejected. I will oversimplify it for you. So when we say a primary endpoint, means this is the outcome that will answer the primary or the most important question. What is your research question? What is the most important questions you want to answer when you do this study or this clinical trial? So this will be the primary endpoint. And when we talk about secondary endpoint, you can have other questions. So I want to know, for example, my primary endpoint will be, I want to know whether this medicine is good, or it's effective or not effective or whatever. Then I can have other secondary endpoint, like what are the adverse events with these medications, what is the rate of hospitalizations, what is the rate of death on therapy, whatever. I can add secondary endpoints. So primary endpoints is answering the main research question. Secondary endpoints are other questions which you may have it with regard to this kind of disease, and you want to answer it, but it's not your main concern. Then we have different types of endpoints. The most commonly, which people knows, maybe the death from disease cause. This is the most common which we use. But the others we use also the occurrence of disease signs or symptoms, because some disease, the aim of the treatment is just to control the symptoms and signs. The aim is not to cure. Some kind of diseases are not curable. So we are just talking about the occurrence, the control of symptoms, and this is in palliative setting. Sometimes the occurrence of disease signs or symptoms. I just gave examples a few minutes ago about sickle cell disease, where patients are presenting with painful crisis, what we call vaso-occlusive crisis. It means they can have frequent pains. So some medications in sickle cell disease, their primary endpoint or the main endpoint is to control the frequency of symptoms, which is the pain and visiting to the emergency. It can be about quality of life, and it can also test the endpoint can be binary. It means it indicates whether an event such as death, whether it's from any cause or not. You have these different types of endpoint. Example, this is not an exclusive. So what are the barriers for in clinical trials? Clinical trials, as I said, it's a complex process. and it requires a lot of, you need to understand the basics of clinical trials, you need to understand the GCP, you want to go through the regulations, but whatever, even if you do all of this, still we have barriers worldwide. Some of the studies focusing on these barriers and found that 70% of the clinical trials fail because finding eligible patients that meet study inclusion and exclusion criteria. And our colleague new to clinical trials frequently falls in these problems. He will come and tell you, I want to do a clinical trial. What is the sample size? He said it was calculated and found to be like, 3,000 or 5,000. And then this number is very big. And it's not only very big. From our clinical practice, we noticed that only 30% of your patients will agree to join a clinical trial. It means if you have 10 patients, 7 of them either they will refuse or they will be reluctant to join clinical trial. Only 30% of those patients they were required to go in clinical trial. Another factor which is also determining the success or failure in the practice is the number of scheduled and visit. If I want to join your trial and you ask me to visit you frequently, you told me, okay, Dr. Yassin, I want you to join a clinical trial. I said, yes, okay, welcome. How frequent I need to come to do this trial? You told me you have to come like 20 times per year. Oh my God, I'm working. I cannot get 20 times out of my work and visit you frequently just to do your trials. And it's a voluntary process. So the number of visits and the frequency or the distance. Some countries are very big countries like US or China. If somebody is living in another province, it will be very difficult for them to travel to long distance, maybe 3 days or 5 days or more than 6 hours by plane just to come to the site of clinical trial. So this is another difficulty. So there are many difficulties in practice. These are the 10 most common. I give 2 examples of them. The third example, which we also face very frequently in our institute and other institutes, is the time taking for ethics or for the approvals. Some institutes are fast, some institutes are slow, and some institutes are extraordinarily slow. So some institutes, they can issue the approval for you within 2 to 4 months. In some institutes, it could go to 1 year. And this is one of the causes of failure for clinical trial, because particularly when you do it with the pharma wants to know exactly when you are going to, they want the time frame. When do you expect to get your ethical approval? I expected to have it after 2 or 4 months. But if I didn't get it after 2 or 4 months, I may be out of this trial because they have a specific timeline. So I hope that I give you an overview. I know clinical trial is it's very interesting, but meanwhile, it's a very complex subject. It's very difficult to cover all the aspects of clinical trial in just 1 hour. And the aim of this presentation was just to touch base with you about the basic concept of clinical trial. I hope that you grasp all what I said, or some of what I said. And I think in the practical session, Dr. Prem will take you further into further details, and you may have a better understanding. Thank you very much.
Thank you very much, Dr. Mohammed. Can I ask you to stop sharing the screen and
Yes.
Dr. Prem, now you should be the host. Okay, Dr. Prem, you should now be able to share the slides from your screen. You'll need to unmute your microphone as well.
Can you see my screen now? Okay, great. Can you see my screen now?
We can see your screen, yes.
So shall I start or should I wait?
Yes, the audio is not very clear. Are you using the microphone?
Yes.
It might be best, yeah, if you take the microphone out and just try using the laptop.
Is it better now?
Yes, that's better. It's clear now. So you may begin.
Is it better now, Dr. Jack?
Yes, that's much better. Yes, thank you.
Okay. So shall I start now?
Yes, yes, please. Yes.
Okay. So a very good evening to all of you again. And first of all, I would like to thank very much Dr. Jack and Dr. Hajam for inviting me to speak on this topic, today's topic on design and appraising the randomized clinical trial studies. And it is always a great pleasure to interact with young researchers and share my research ideas and experiences on this important topic. And certainly, it could have been better to present this in person, but unfortunately, due to this current circumstances, I think this is the best possible way that we can discuss the topics. Okay. So I'm unable to. Yeah. Okay. So in my next 45 or 15 minutes talks, I would be discussing mainly about how to make a critical appraisal. So to start with, kindly note that because of the time limits, we would be talking about a very basic concept about different steps that we used when we do a critical appraisal of RCT, particularly RCT studies. So this is a very brief outline about today's presentation. As we have already discussed about understanding the significance of randomized clinical trial study design. Previous speaker has spoken about this, and we will also be discussing about learning why do we need to perform a rigorous critical appraisal of RCT studies, and we will discuss about understanding the basic principles of critical appraisal and its role in evidence-based practice. And I hope that the end of the session, it would help you and to appraise the research and judge its reliability and validity, and of course the relevance of the research to your own work. Last but not the least, we will also be discussing on a very lighter note about the different standard guidelines and resources which are available to report and critically appraise cities such as CSV, Consort, and Equator Network. Okay. So before I go and present the main topic, I would like to preface some of the statement which was published in BMJ by Professor Douglas Altman. And many of you might be knowing that Professor Douglas Altman is an outstanding statistician and a leading scientist, particularly worked in improving the quality of research, particularly in methodological point of view, where he developed enormous number of guidelines, including Consort and many other guidelines which are there on the Equator Network. In few minutes, we will discuss about those guidelines very briefly. So the point which I would like to highlight here, what is said in that publication, the title, it was like what he said was, we need less research, better research, and research done for the right regions. And further, what he said, abandoning using the number of publications as a measure of ability would be a start. So what he meant to say is, we need to focus on quality of publications, not about the number of publications. So, and then he said that to maximize the benefit to patient and society, you need to not just do a research, but do it well. And this is the most important message that he conveyed to all the researchers who are working in the field of biomedical research. And final statement was like, I would like to preface one of the more statement that given by another author published in the journal. What he said was, accurate, transparent reporting is like turning the light on before you clean up a room. It does not clean for you, but does tell you where the problems are. So this is exactly what we need to do when we do a proper critical review appraisal of any RCT. Not only RCT, but it applies to other observational studies too. So the whole idea is we have to be very clear about what method that has been used in order to conduct RCT design. When I say method, it includes many components. And this is the slide which is showing, if you look at here, the scientific method sits center of everything. When I say method, so we are talking about hypothesis formulation, we are talking about design, we are talking about creating and implementing the data collection set, and we are talking about data analysis and statistical testing, and we are talking about interpretation of findings. So if you see it here, the scientific method really sets the foundation for any studies. So this slide is basically showing us the different level of evidence that we have, which are coming from different study design. If you see it here, the case report, which sits at the bottom, whereas systematic review and meta-analysis sits on the top, and next to it is RCT. So we all know that RCT is probably one of the best designed to create a highest possible level of evidence. So we have to be very careful about when we plan, design, conduct, and analyze RCT studies. And the Prayasin has already covered those things. Why do we need to have a sound RCT design? And I just would like to touch a couple of advantages of having RCT design. We all know that this is the design which is considered to be the methodological standard of excellence for scientific experiment, probably one of the best design for controlling a selection bias. And it also controls the confounding bias without adjustment. In a minute, we'll get to know that what we mean by confounding. And I'm sure that in the presentation on topic qualitative studies or observational studies, the presenter might have discussed about the concept of confounding and bias. So I'm not going to discuss in detail, but I'm sure you all know about what exactly the meaning of confounding and what exactly the meaning of bias. So keep in mind that this study design would take care of this two component very effectively. And it is also one of the best measure of causal relationship. And I'm sure when the presenter discussed about a cohort study, you know the cause and effect. So like exposure and outcome, exposure and disease. So probably in this design, this is one of the best design where you can measure the causal relationship, which causes what. Does the intervention causes reduction in the pain, or does certain, let's say physiotherapy causes a better quality of life, better, So you can really assess the cause and effect relationship. And it can also measure multiple outcomes. When I say multiple outcomes, as Dr. Yasin pointed out, in RCT, there could be one or two primary endpoint, and there could be a number of secondary outputs, endpoint. So when you conduct RCT, you can focus on one or two main outcomes, which we call as primary outcomes. And remaining can be classified as secondary outcomes, which can also be addressed using the same study, RCT study. So in one study, you can address both the primary outcome and secondary outcomes. What are the certain disadvantages? It definitely involves very high cost, ethical issues. The previous speaker has already spoken about, maybe a problem sometimes, and the compliance is one of the biggest issues in RCT study design. We have to make sure that to maximize the compliance so that we can really generalize the conclusion of the study. The question is, are the RCT results always valid? This is the question. So RCT can provide a conflicting results. RCT design, executions, or analysis can be flawed. Intervention versus control comparisons are internally valid, but restrictions on participant visibility can reduce external validity. So not always the RCT designs provide a valid evidence, valid conclusion, unless it was conducted with proper methods, with proper planning, with proper steps that are needed from the ethical point of view. So the studies which are conducted as RCTs, if it is not actually conducted as RCT, will never provide very sound evidence. So what we need to make sure that all the researchers must need to use standard guidelines applicable in conducting and reporting high quality RCTs. In few minutes later, we'll get to know what are those standard guidelines which are applicable to conducting and reporting randomized clinical trial studies. Okay, so here is a question for you. In an experiment to compare two treatments, advantage of random allocation include. So you need to answer either true or false. So this includes the sample may be referred to a known population. So you need to answer is true or false. The experimenter will not know what the treatment the subject will receive before recruiting them into the study. And the C, option number C, the likely size of errors can be estimated. The option D is the two groups will be similar apart from the treatments. So the treatments are assigned according to the characteristics of the participants. So you need to answer for each option, you need to answer whether the answer is true or false.
So, Dr. Prem,
Yes.
The participants will see the poll and they will indicate the answers that are true. So in about 15 seconds, you'll receive the poll results and you will see the responses that the participants have deemed to be true.
Okay. Okay, so I can see it here. Three of the participants said the B, which is the experimental will not know. Yeah, that's correct answer. And the two of them said C, which is again a correct answer, and 5 of them said D, which is again the correct answer. So I can see the 10 out of 36 participants answered correctly. Okay, I think that's great. So now we can move to the next presentation.
Yeah,
Next slide.
just click X on the poll results. So X in the top right hand corner. So you'll need to close that because it's forming a shadow on our screen. So you just close that one.
Yeah, this one.
Yeah. Perfect. Okay. Thank you. Thank you.
So now, after we had discussed very briefly about why do we need to have critical appraisals, now we will get to know that what exactly is the meaning of critical appraisal. What is critical appraisal, first of all, and how does it differ from standard guidelines? So in a very layman, in a very simple terms, the critical appraisal is basically a systematic process used to identify the strength and weaknesses of a research article. So the bottom line here is the strength and weaknesses of research article, mainly focusing on the methodological part. Is the method reported there in the trial is adequate or not? That's what exactly we do when we do critical appraisal. So the critical appraisal provides a basis for decision on whether to use the results of a study into clinical practice or not. Because we all know that if the methods are not proper, if it is not adequate for the design, definitely the evidence cannot be used in the clinical practice. So the different design are prone to various sources of bias and confounding effects. And therefore, we must need to use the critical appraisal checklist with design specific to assess the study quality. So I know that there are different checklists for the different types of study. For the observational studies, we have a case control study, we have a cohort study, we have a cross-sectional studies. So for each study type, there are the different critical appraisal checklists. Likewise, for the RCTs, we have the different critical appraisal checklists. So make sure when you do a critical appraisal, look at the appropriate checklist which are applicable to that design, whether it's RCT or other study design, and make sure you use the proper checklist applicable to that study design. So, and then the last but not the least, assessment of other factors like including the importance of the research question, the appropriateness of statistical analysis, the legitimacy of conclusion, and potential conflict of interest are an important part of when we do a critical appraisal. So make sure it is not about the method. It is not only about the method when we do a critical appraisal. There are a lot of other factors that we need to take into account to judge the quality of the study. Okay, so the next one is about why we need to perform a critical appraisal of RCTs. So okay, so the critical appraisal is an essential skill for evidence-based practitioner. It involves the systematic examination of research in order to assess its trustworthiness and relevance to specific situations, as we all know that. And the critical appraisal, which aims to ensure that unreliable evidence is not translated into practice. That's most important. Like the evidence which are coming from the best quality studies should be translated into practice. Otherwise, it may lead to very potential harm to patient and of course the wastage of resources and money. So this is why we have to be very careful about judging the validity and reliability of the evidence which are coming from RCT experiment. And that is why we need the critical appraisal to make sure that the proper method has been used, the proper steps has been used, the proper planning had been placed so that the evidence can be derived with a very sound quality. And randomized controlled trials are, we all know that, are regarded as the gold standard for evaluating the effectiveness of intervention. And I'm sure you all know that the difference between effectiveness and efficacy. So efficacy, when we talk about efficacy, we talk about single trial. So we define the criteria, we conduct the study, we talk whether the one drug is more efficacious than other drug or not. When I say effectiveness, if I'm talking about something about a general population. So there is a slight differences between the two terminology, which we call effectiveness and efficacy. So the critical appraisal of randomized controlled trials relies heavily on the clear and transparent reporting, the design, conduct, and analysis. I will repeat this last sentence. The critical appraisal of randomized trials relies heavily on the clear and transparent reporting of the design, conduct, and analysis. This is why we need the guideline to report a clinical trial. And the guideline which we use is called consort guidelines. We'll discuss very briefly about what exactly the consort guidelines is and why do we need to use the consort guidelines to conduct and report properly a clinical trial. So now, what are the main key components under the critical appraisal? So basically, there are three components which we look for when we do a critical appraisal. We look for internal validity, we look for biases, and we look for external validity. I'm not going to discuss the detail about the different types of biases that we have called performance bias, detection bias, attrition bias, reporting bias. I'm sure that the previous speaker might have spoken a detail about the bias, confounding, and errors. So I'm sure you all know about the differences between the bias and the error and the confounding, which really significantly impact the study finding. So we have to be very careful about how are we going to minimize the bias? How are we going to identify the bias? what technique I should use to minimize the bias, to maximize the quality of the evidence, findings. So we have to take care of this. And when we do a critical appraisal, we look three components very closely and very appropriately. Okay, so what are the different models of critical appraisal? So basically, there are three models that is basically used. And these are the scales, checklist, and domains. Due to the time limit, I'm not going to discuss each one of these. However, the 2 days I will be focusing my talk on the critical appraisal using the checklist. Of course, the initial model was mainly scales, which is now not being used frequently, and there are a lot of drawbacks using the scales to do the critical appraisals. And there is another one called domains. When we talk about the domains, these basically focus on very specific element of the study design, conduct that might adversely affect the internal validity of the study. So these criteria can differ depending on the review question and topic. So again, it's slightly subjective. Rather, this tool, basically the domain tool, assign a risk of bias for each domain, such as randomization, and consider what the study has reportedly done to minimize that bias. So look at, this is very clear that we put the kind of risk of bias for each domain, like randomization, sample size, those kind of things, and consider what they have done to minimize that bias. So there is a differences between the three models: scale, domains, and checklist. Checklist we'll discuss in detail that. Okay, so as I said before, there are the various tools, checklist, domains, and of course scores, scales. So when I say checklist, there are the many checklists like CASP checklist, Cochrane risk of bias, we call ROB2, Center of Evidence-Based Medicine, we call CEBM, the National Institute of Health, etc. So these are the models which comes under the checklist. In addition to that, the scale, when we say scale, there is a Jadad scale, Delphi list, Chalmers scale. I mean those kind of scales which are not commonly used or recommended nowadays. So these are the different tools for guidelines and the critical appraisal. We are going to discuss today about CASP, Critical Appraisal Skills Program, and there are the two guidelines, and we will discuss very briefly about what exactly the guideline means and what is the difference between the guidelines and the critical appraisal. Okay, so why all are okay, so give me a second. Yeah. Why guidelines? So the publications are basically intended to advance scientific and medical research, healthcare practice and standards, and most importantly, patient quality of life. And the guidelines basically helps establish or reinforce the best practices for healthcare institution to achieve these goals. How to achieve the goals? Develop unbiased data-driven publications with the best quality or best methods used, and provide the full transparency of the data as well as authorship and contributors, and document that all activities are to the highest standard. So it is clear that when we say about the guidelines, we need to make sure that so the design, plan, conduct, and reporting all in accordance with standard guidelines which are needed for that particular study design. And currently we are discussing about RCT. So we must need to know what are the items that has to be reported when we conduct RCT design, because our goal must remain excellence in all our research and publications. As we all know that advances in healthcare and patient lives and safety depend very much on it. Okay, so this is the slide I was talking about. that we do have the different guidelines. If you can see this one, we have a consort guidelines, which stands for Consolidated Standards of Reporting Trials, applicable for randomized clinical trial study. We have a stroke guidelines applicable for observational studies, which could be case control, cohort, cross-sectional. I'm sure that in the previous talk, the speaker might have spoken all about in detail. And we have the guideline called PRISMA, applicable for meta-analysis, STAR, and so on. So the question is, there's so many guidelines, so little time. So what to do? We have plenty of guidelines and very little time. So what to do? Follow the legislation as it implies to your institution. Follow the reporting standards relevant to your data set. Look at what are the key components, what are the key standards that I must need to use when I conduct such a specific type of RCT. When you conduct RCT in, let's say, in the chronic disease, when you conduct RCT in other therapeutic areas, make sure that you include all the components which are essentially required to report and conduct RCTs in that particular therapeutic areas. So, and review the ethics statement standards issued from professional organization. When you encounter a gray areas, let the following goals guide you: integrity, completeness, transparency, accountability, responsibility. I think that applies not only to RCT, but in all types of research. And we must need to follow those standards when we plan, design, conduct, analyze, and report not only RCTs, but all types of studies. And the good publication practice basically helps to advance the science and medicine and demonstrate our commitment to patients, scientists, and healthcare professionals. So, as I was discussing before, that what is the difference? The question for you is that what is the difference between guidelines and critical appraisals, particularly in the context of RCT studies? So if you can throw some light on it, what exactly you feel that this could be the main difference between the guidelines and critical appraisal. Here is the answer for this one. So reporting guidelines is basically to ensure that the full information is provided about study design, conduct, methods, and findings. For the researcher, the guidelines guides you how to report the study. And the reviewer is basically used it to assess the adequacy of reporting. Whereas critical appraisal, if you look at, it is basically like for the reviewer and readers to assess the adequacy of study methods. So look at the differences. In the guideline, we focus on transparency of reporting, how to report the study, and assess the adequacy of reporting. Whereas in critical appraisal, we focus more about the methods. We look at whether the study really follows all the required method, which has to be used in such a specific RCT study design. So it's all about the methodological point of view, how robust your method is, how robust you had used all the steps from the methodological point of view, which essentially had to be used while conducting, planning, designing RCT studies. So the clear differences is that the one in the guideline, we focus more on reporting, and whereas in critical appraisal, we focus more on evaluating the methods which had been used in that particular study. So it cannot assess the method used if those methods are not described in adequate detail. This is what is the significance of following the guideline. If the author failed to demonstrate, failed to mention the detail about the methods, it would be practically impossible for any reviewers or readers to judge or assess the adequacy of the method. So transparent reporting is a must, is a prerequisite criteria to do a critical review appraisal. And good reporting is necessary for judging the study methods about on relevance and reliability. Without without having adequate reporting, it would be next to impossible to judge the method. So this is why I again and again focus on, when you plan any RCT study, make sure you start the day you start the planning, the day you start the designing, the day you start the study conduct. So all the steps has to be very well planned in advance, because RCT study design, our planning has to be very perfect. Our steps in the design has to be very, very, very adequate. And we all know nowadays in the journal, most of the article RCT studies got rejected, not on the ground of analysis and reporting, but on the ground of design itself. Though the steps were not good enough to accept the study as an RCT. So this is why the reviewer decided to reject the RCT studies, because it wasn't meet the criteria for qualifying the study as an RCT. So we have to be very careful about the steps which are being used in the plan, design, and conduct before we go and start collecting the data. Because study foundation lies in how we design the study. Did we follow the proper methods or not? If not, your data quality is going to be questionable. So we have to be very careful about that. And this is why I again and again would like to focus on, why do we need to be very, very particular about when we plan, design, and conduct the study. Because once the study is done, you cannot go back and correct the things which were not accurate at the design stage, not accurate at the planning stage, not accurate at the data collection stage. So this is the point which I would like to highlight it here. So before we go and talk about it, I just would like to show you here the one of the editor reports of the manuscript problem. You can look at here. It's quite clear that the top three, which is the poorly written, excessive jargon, inadequate, inappropriate presentation, and the poor description of design, the three components which commonly, frequently occurred in manuscript, and that is coming from RCT study design. So we have to be very careful about those. And two other components, which are the sample size and analysis method. Many of the studies really lacks providing the details about those two components, which are extremely, extremely essential. Sample size, I'm not going to discuss in detail, but sample size is an absolutely a very critical part in any RCT study, not only RCT, but observational study too. Having less is not good, having more is not good. What is good is having adequate. And don't worry about how to compute it. You go to statisticians or epidemiologists, he or she would help you how to compute that number based on what exactly your primary outcomes are. Okay, so this slide is basically showing you the frequency of poor reporting inadequate methods in RCT studies. That was published in BMJ by Agnes and et al. So he analyzed the data of more than 20,000 RCTs published in more than 3,000 journals. I just wanted to highlight the reporting, inadequate reporting of different component. If you look at here, the proportion of trial with unclear risk of bias was 40, almost half of the percent, 50% for sequence generation and 57% for allocation concealment. And the proportion of those with high risk is 4 and 7.2% respectively. Look at the frequency. It's close to 50% and more than 50% regarding sequence generation of randomization and allocation concealment, which we know is the very fundamental part of any RCTs. And it is almost 50% of the studies where they did not report this very, very clearly. And for blinding and incomplete outcome data reported in 31% and 24%. Again, it's 1/3 of the proportion. It wasn't clear. And then the proportion of trial at unclear risk of bias decreased over time, which is a significant finding, especially for sequence generation, which fell from 69% in 1986 to 90 to 31.2% in 2011 to 14, which is a great achievement, I would say, because bringing down from 69 to 31%, more than 50% reduction, which is great, great achievement. So And similarly, for allocation concealment, it came down from 70% to 44%. So in summary, poor reporting and inadequate method have decreased over time, especially for sequence generation and allocation concealment, but more could be done. Because that's not only the two components which basically sets the foundation for RCT, there are many other components. So we have to carefully look at those steps as well to make sure that it has been followed properly. Okay. So after having discussed very briefly about the guidelines, very briefly about why do we need to have the critical appraisal, and very briefly about the frequency of different types of what you call errors that was found in RCT studies. We will now discuss about, as we had discussed before, about critical appraisal checklist, which is CASP, which is the main checklist that we are going to discuss today. But please note that there are many other checklists which are there for the for critical appraisal, for particularly for RCTs, but we are going to focus here with the time limit. We are going to focus on CASP. So CASP is basically having a 4 section: Section A, Section B, Section C, and Section D. Section A is basically talking about the basic about the study design, whether it's valid for randomized trial or not. Section B is about the methodological point of view. Is the study methodologically sound or not? Section C is about the results and its validity, and Section D is about whether the results help locally or not. So this is the platform where you can download all the checklist, not only for RCT, but all the other studies as well. I'm not going to discuss in detail, but look at the screenshot here. The checklist is available for randomized clinical trial, case control cohort, and those kind of studies. So these are the 11 items that we have under the CASP checklist. And we will discuss in a minute briefly about one by one. So the first component is all about, was a clearly focused research question being asked or not? Were the patient randomized? Were all the patients who commenced the trial accounted for conclusion or not? Were participants, staff, and researchers blinded to participants or study group? Were the groups similar at the baseline? Apart from the intervention, were groups treated the same way? What were the main results? How precise were the results were all important outcomes assessed? Can the results be generalized to the local population? Last but not the least, do the benefits outweigh the cost and harms? So we will discuss one by one in detail. Dr. Jack, can I click on here and open this one, or should I open from my desktop?
I think you'll have to open it from your desktop and share the screen from your desktop.
Let me minimize this and I will open through my desktop here. So I can share in the same way, right?
Yes, so if you open the document and then you can share the screen with the document.
Okay. But I'm unable to see the. Should I first stop the sharing and then share it, or how to do that? Because the PowerPoint is still open here.
Okay, if you press share now, if you click on share.
I'm unable to see the. Unfortunately, I'm unable to see the. Dr. Jack, I'm unable to see the share screen. Okay. Unfortunately, I'm not able to see the. I can only see my. Should I first stop sharing and then say, because PowerPoint is...
Yes, you can try that. So if you stop sharing and then try to share from the PDF.
Okay. Now let me share the... Yes, here. Can you see it now?
We can see it.
Dr. Jack, can you see it now?
Yes, we can see it. Yes.
Is it visible?
Yes.
Okay. So I was talking about the different checklist and the different items that are there in the CASP checklist, which we call critical appraisal skill program. If you look at the item number 1, so section A, item number 1, did the study address clearly a focused research question? So you have to look at, did they really formulate a research question using the concept called PICO, which I am sure you know that the PICO means population, intervention, comparators, and outcomes. So you need to review it properly and see whether the question really formulated based on this criteria of formulating a good research question, which are coming using the term called PICO, which is called population, intervention, comparison, and outcome. So the second item is about what the assignment of participants to intervention randomized. So it is important to know, it is important to review it, how was randomization carried out? Was the method appropriate or not? Was randomization sufficient to eliminate systematic bias? Was the allocation sequence concealed from investigators and participants? So we need to be very careful about how did they do a randomization? Did they put a sufficient detail about how the randomization was done, who generated the sequence, who implemented the sequence, and who allocated the treatment to patients? So all the details had to be there in the research. And the third one is where all the participants who entered the study accounted for its conclusion. So it basically tells about the loss to follow-up and exclusion after randomization. So we have to look at very carefully how they address about loss to follow-up. Did they use any kind of method to take care of this, or the loss to follow-up, we need to look at what is the percentage and how does it affect the study finding. If it is a very huge percentage, definitely the study will be underpowered. We have to be very careful about that component. The section B is about where the participants blind to intervention they were given. So we have to look at section B is all about to assess the methodological standard about the blinding, and where the people assessing the analyzing the outcomes are blinded. So we all know that what is single blind, double blind, triple blind. We call it a triple blind, where the one who is going to analyze the data is also blinded. So like statisticians, he may be getting data in terms of intervention A and B without knowing what exactly A, what exactly B is. So we have to be very sure about the blinding assessment. Was it single blind, double blind, triple blind, or was the detail provided in the research manuscript or not? And then we have to look at very carefully where the study groups similar at the start of the randomized trial, because we all know that when we do a randomized study, when we do a random allocation to the participant, it is very fair possibility that we would unlikely to have imbalance at the baseline. Most of the factors are going to be balanced at the baseline. When I say balance, it doesn't mean that you'll have equal distribution. You will not have, let's say, 50% male in one group and 50% male in another group. It is not expected. But you may have 50% versus 48%. Or you may have, like comorbidities, 30% versus 28%. Quite same in both the treatment groups. If it is not, which really tells about the validity of the randomization. Did the author really did the validate what you call randomization properly or not? So when you do a proper RCTs, it is quite obvious that the most of the factors that you measure at the baseline, the covariates, will more likely to get balanced apart from intervention. So you need to assess that one too. Because, and this is the one thing where we say randomization not only controls the known factor, but also it controls and adjusts the unknown factor which we don't know. So imagine that there are some covariates or factors which are affecting the outcomes later on. And unfortunately, you did not collect it at the beginning. It is more likelihood that even if you would have been collected, those parameters would have been balanced. So this is why we say randomization does not only control known factors, but it does control unknown factor as well. So the point number 6 is all about, apart from the experiment intervention, did each study group receive the same level of care, which is very important. I mean, we have to be very, this is where it might lead to something called detection bias. So if the different care was provided for the different group, let's say you are comparing treatment A and treatment B. Let's say if I am the investigator, I know that what exactly the treatment A, and then I might be giving a different kind of care for the participant who are in the treatment group A than treatment group B. So this will really affect the outcomes. So we have to be very careful about that. So the care should be same for both the groups. And then, so from question number, if you can see from question number 1 to 3 belongs to section A, which talks about the study design validity. Question number 4 to 6, it talks about the methodological point of view, about the randomization generation, about the balance at the baseline, and about the care provided in both the groups. And then question number 7 is about validity of the results, where the effects intervention reported comprehensively. So we need to make sure that your results should be in line with the objective that was there at the beginning of the study. How about the primary and secondary objective, how the results are reported, and adequacy of statistical method has to be assessed appropriately. So this is why they said, like here, if you see it here, was the differential dropout between the study groups that could affect the results, where the potential source of bias identified, which statistical tests were used, where p-values reported. So those things are needed when we talk about comprehensive findings. Number 8 is about what the precision of the estimate of the intervention treatment effect reported. When I say precision, means we are talking about 95% confidence interval. I am sure you all know that when we report a p-value, we must need to report a 95% CI as well to measure the precision of the estimate that we get it. And then item number 9 is all about, do the benefits of the experimental intervention outweigh the harms and cost? So this is called risk-benefit ratio that we must need to ensure when we look at and do a critical appraisal. So the number 10 is all about, can the results be applied to your local population? You have to look at the validity of the results in context with application in the local population. And then the last one is about, would the experimental intervention provide a greater value to the people in your care than any of the existing intervention? So all need to be very critically assessed when we evaluate a critical appraisal. So if you look at all 3 sections, basically talking about the design validity, is the RCT is the best design to answer that question? The second step is all about assessing the methodological steps, and third step is all about assessing the results validity and reliability. This is what exactly we discussed about at the beginning. When we talk about critical appraisal, we talk about internal validity, we talk about bias, and we talk about external validity. So this is exactly what we look for when we perform a critical appraisal checklist. So now I will go back to my previous PowerPoint slide here. So again, I need to use the Dr. Jack, can you see my screen now?
Yes, we can see your slides. If you can put it to presenter mode, yeah.
Yes. Is it visible?
Yes, Doctor.
Yes. So now this is a question for you all. So the question here is, in a randomized double-blind trial to compare a new analgesic with placebo for the control of pain in arthritis, subject reported less pain while using the analgesic, and the differences in pain score between the two regimens was highly significant, which is 0.002. We can conclude that, number A, you need to answer whether it's true or false. So number A is an important clinical advances that has been made. The B, there is a good evidence that the drug reduces the pain. C, the drug is a very effective analgesic. And number D, it is unlikely that the results would have been obtained if the drugs were totally ineffective. And the difference between mean pain score on the two regimens was 0.002. So you need to answer each one either true or false.
The participants will need to indicate which ones are true. So the participants will need to indicate which of the options are true.
Right. So
available to them, they will have to indicate which ones are true.
Yes, which, like, for each one, they need to answer which one is true. Like the first one, let's say either is true or false. Second one could be true or false. Third one, so for each option, the participant had to answer in terms of either true or false. Okay. So now we'll see that what is the response here.
Yes, we're just collating the results, and in 10 seconds you'll have the scores.
Sorry, Dr. Jed.
You have the scores.
Okay, so I could see here. I assume that the one who said here is in favor of true, right? Like I can see there is a good evidence. The number option B, 8 of them said true. I believe they are opting the true, right? So like 8 of them said, because the answer could be true or false. So I'm going with the, like, once they say that it is true. The B is like there is a good evidence that the drug that reduces the pain is true. And the D, it is unlikely that the results would have been obtained if the drugs were totally ineffective. That is also true. So A and D are true, whereas, B and D are true, whereas A, C, and E are false. So I assume that, I could see that 6 participants said true for D, and then 8 said true for B, which is good. Okay, so now we move on to our next slide here. Yeah. So after having discussion with
Dr. Prem, you
yes.
need to close the polling. Click on the X, yeah.
So after having discussion with about very briefly about the CSP checklist and the critical appraisals, I know that we are running a little fast due to the lack of time, but I'm sure that if you have any questions, so please feel free to contact us, and we would be happy to help you in order to know the detail about each of the component. But in the limited time frame, I think we are just touching about the concept, about the component. But to know, we all know that every component requires 3-4 hours to explain the detail about a critical appraisal. So keep in mind that this is just about the basic concept of why do we need to have the critical appraisal and what is the difference between critical appraisal and guidelines. So the final section is all about the guidelines, as we had discussed in previously. the consult guidelines which are applicable for RCTs. We are not going to discuss in detail, but my here aim is to show you that what are the checklists, how many items are there. If you look at there 25 checklists, so from item number 1 until item number 12, it's all about the study plan, design and methods. From item number 13 it starts with results. So when you design the study, look at your protocol really includes the item number 1A to 12B. So use that guidelines, and I'm sure when you use it, you will definitely plan, conduct, and report high quality RCTs, and you can easily get it published in a very, very good journal once you start following guidelines right at the beginning. Do it at the day you plan the study, not the day the study is done, because anything flaw in the design, anything that really leads to improper design will be extremely difficult for anyone to accept the evidence. So this is the checklist. So this is the detailed checklist items. Whenever you have time, please go through it and feel free to contact us to know more detail about each component. So the third question for you is, in a randomized double-blind trial to compare a new analgesic with ibuprofen, standard treatments for the control of pain in arthritis, the difference in the pain score between the two regimens was not significant. Can we conclude that the drug is useless? And there is no differences in analgesia between the two drugs? The trial has failed to demonstrate a difference in analgesia, and the difference between the drug is very small. There are no important differences in the analgesic properties of the drug. So you need to answer either true or false. So each one of the options could be either true or false. So can you provide us your feedback? And this should help you when you start reviewing the result section of any RCT article. You get to know that whether the results are really consistent with the design, with the method, with the conclusion. Okay, so now the poll has ended. Let's see the response. Can we have the response, please? Yes, I can see it here. The question C, which is the maximum number, I'm happy with that because this is the right answer. The trial has failed to demonstrate a differences in analgesia. Excellent. I think I am very happy with the response that I got it. So this basically shows that, because remember that the p-value, when you get the p-value which is not significant, does not mean that there is no differences. The only thing that we can say, there was not a sufficient evidence that to show that there was a difference, but we cannot say that there was no differences. Like we cannot say that the new drug is useless. No, we cannot draw this statement having the p-value not significant. We cannot draw the statement saying there is no differences. There was no statistically significant differences, but it doesn't mean that there was no clinical differences. So always keep in mind that when we talk about significant p-value, there is always a two significance: clinical significance and statistical significance. So always relate both components together. The study may have clinically significant, may not have statistical significant. Other way around, the study findings may have clinically significant, statistically significant, may not have clinically important. So keep in mind that component when you start interpreting the study finding. So I think to summarize it, we all know that Randomized controlled trials that are well designed, well conducted, provides the most reliable evaluation of the effectiveness of healthcare interventions, and trials that are inadequately conducted and reported, however, could result in significant bias and exaggerated treatment effects. So we have to be very careful about that. And it is important for medical practitioners to be able to critically appraise randomized control trial in order to prevent such biases, which really played a very inverse role in misleading the clinical decisions. And using the 4 steps that outlined in the critical appraisal checklist today might provide a very useful framework for this process. And keep always in mind that today's research is tomorrow's medicine. So one point before I close this, I would like to highlight, when you plan RCT, plan it, your planning success depends on the way you plan, the way you design, the way you place your method, the way you start collecting the data. Once your data had been collected, your most of the tasks gets over. Now we have the beautiful statistical software where the analysis can be done very quickly and reported very quickly. So make sure when you have all the steps in place, your good plan, good design, good steps, methodological steps, that will definitely lead to a good quality of the data, and that will definitely lead to a good research outcomes and publication and evidence that really will affect the patient quality of life. And this is what exactly the main aim of doing a good quality research, not only RCTs, but even the observation study too. And thank you for your kind attention and listening. So I hope that, I know that it was a little faster. However, the topic is very wide, and anytime I know that you may have multiple questions. So feel free to approach us, to contact us, and we would be happy to help you as much as we can in order to discuss the different research ideas, not only about RCTs. So within this limited time, I think the idea was just to take you through the basic steps, but for details, you are more than welcome to approach us and contact us. Thank you so much.
Thank you so much to our speakers today, Dr. Prem, Dr. Muhammad, and Dr. Hassan, who is also involved in developing the content today. Thank you to Mr. Shuja and our logistics team and Ms. Saha for arranging today's event. And thank you as well for attending and taking part. We very much appreciate you coming along. And please provide your feedback to us. So next week we'll be starting our qualitative research event. So we look forward to welcoming you next Tuesday to our first of 2 events on qualitative research. Thank you once again, and good night. Sweet dreams, everybody.
Thank you so much. Good night.
Thank you.
Good night, everyone.
Good night, everyone. Okay.